Yeah.
Dear participants, today in this technical session, I'm blessed to have with us one eminent and renowned scientist in the field of drug delivery, Professor Biswajit Mukherjee, who is my teacher at Jadavpur University, Kolkata. When I left Jadavpur in 2008, that time sir was in his peak of professional career. And today, after a span of 12 years, still he is the best in the field, which speaks a lot about his character. He is such a dynamic person. He is so active that it is difficult to match up his energy level. I tried very hard, but it's really difficult, I must confess. Let me read out a brief bio sketch of Sir. Sir is presently working as professor in pharmaceutics in Department of Pharmaceutical Technology, Jadavpur University, Kolkata. He is former DAD fellow from German Cancer Research Center in Heidelberg, Germany. Additionally, Sir is holding various academic positions. He is the coordinator of AICT-sponsored Quality Improvement Program, that is QIP, Nodal Cell of Pharmacy, and Joint Coordinator, Center for Advanced Research in Pharmaceutical Technology Department of Jadavpur University. He is the convener, Animal Ethics Committee of the University, and the coordinator, Department of Science and Technology, it's Government of India, PARSI Program 2 of the department. He is also a former faculty of University Institute of Pharmaceutical Sciences, that is Punjab University, Chandigarh. Sir is an outstanding and renowned researcher in the field of pharmacy. His specialization is antisense technology and chemoprevention in cancer model. He has been working on novel drug deliveries, particularly transdermal patches, nano-size liposome, and neosomal formulation, as well as nanoparticles. Based on his distinct research field, Sir is awarded on an average 4 research grants every year. So meaning that in his 20 years of teaching as well as research career, Sir has handled around 80 research grants. So you can imagine 80 research grants from various government funding agencies such as UGC, CSIR, ICMR, DST, DBT, DRDO, etc. This is of huge achievement, and he has a number of patents in process. He has published more than 150 research articles in many international journals of repute. He has also written 10 books and several book chapters. Sir has supervised 35 PhD students and 72 MPharm students. Sir has been awarded many academic and research awards. He is the recipient of UGC Research Award 2009. He has received some other awards also. He is acting as editorial board member, guest editor of many international journals. Among them, one is the section editor of Encyclopedia of Metalloproteinases, a renowned journal from Springer. Actually, Sir has very lengthy CV and it will take long time to cover. So with Sir's permission, without taking much time, I welcome my Sir Professor Biswajit Mukherjee to deliver his talk. Today, Sir will be talking on the topic drug targeting by nano formulations. It is an interesting topic. I'm very sure that the participant will be hugely benefited by your lecture, sir. So, sir, it's over to you.
Thank you, Tom, for your nice introduction. First of all, at the very outset, I like to convey you belated Dasera, as well as, the forthcoming happy Diwali, because Diwali is just...
Thank you, sir. Thank you.
So thank you to all of you. I understand that this is probably the staff development program, right? Faculty
yes,
development
sir.
program. So the participants are mostly, I think, faculties. Of course, I will discuss mostly on the research areas. And yes, if you never feel any hesitation to ask me questions, You will get the opportunity, whenever you will get the opportunity, you can ask me the questions if something is not clear to you, because by interaction only we can learn. You understand that we are just, you can say, nearly 2,700 kilometers away. From the east to west, we are connecting because of this device, computer and internet. We are so fortunate today that from this extremely eastern part to the almost nearly extremely western part, we are just delivering our lecture and interacting. I was in Chandigarh for quite some time. I was a faculty of UIPS, Punjab University. So I think winter already has started there or just knocking. So in this particular environment, let us discuss about a topic. I will discuss it slowly so that you can understand and you can enjoy it. Do not hesitate to ask any questions whenever you feel clarification is needed. Okay? So should I start?
Yes, sir.
Yes. Can you see my slide?
Sir, not yet.
Now?
Mm,
Not yet.
no, sir.
Okay.
Not yet, sir.
Okay. Can you see
Sir, go
my?
to the share screen.
Okay. Just give me a little time. Okay. Now, can you see my desktop?
Yes, sir.
Yes. So now I think you can see the slide. Yes.
Yes, sir.
Can you
Visible,
see?
sir.
It's okay. And also you
Okay.
can hear me. Okay. So let me start. As you understand that we are going to discuss about dark targeting and of course by nano formulation. So two aspects I am just going to deal with. One is the nano formulation. That means formulation is the carrier, drug carrier, you understand. And carrier means the thing which just carry us from one area to the other. We are not dealing with drug. Our interest is the vehicle, carrier. And second thing is our focus is to target, to just, deliver the cargo at the right location. Cargo here, the drug cargo. So our objective is to deliver the drug cargo to the right location of our body. You understand this is very important. Why it is important? You know that I want to send some materials to New York from Kolkata or maybe from Mumbai or Delhi. And that material may be few tons, 5 tons or 10 tons, but during its, transportation from Kolkata to New York, on the way, maybe 80% of my material is lost. Out of 10 tons, maybe 8 tons are lost and 2 tons are reaching to the target. You understand? Then it is a huge loss of our material, huge loss of our material. So this is happening in our system. We are giving drug because we are unable to localize it to the right direction. Our objective is to put it to the right direction. If we fail, then you know there will be a loss. It is nothing unknown to any one of us. But we are very careless also because in you can say the drug industry came properly in the proper shape in the second decades of the last century. That is around 1920, 1989, 1990, 20 onwards. Slowly it came, but it is nearly 100 years still. We are unable to locate. We are unable to deliver our cargo to the right location. This is the time when our focus is not to take the medicine only. Our focus is just to deliver the medicine to the right location. And that is targeting. That is wanted. That is targeting. So we want to bring our drug to the right location. Little earlier I said, we are not working with drug at all. We are working in the delivery process, transportation process. That is the vehicle. Vehicle is the carrier. Like somebody is standing one side of the river, he wants to go to the other side of the river. So he needs a boat or maybe some other ship or something else by which he can be delivered from one side of the river to the other. So that boat is the vehicle, the ship is the vehicle. So we want that vehicle in which the drug cargo, here the human being, as I said, in case of boat, in our case, the drug cargo can be transported to the other side. But to the other side means to the right location. If I want to send something to New Delhi and if it reaches to Mumbai, it will be, I do not know how I can react. So, but in our body, in case of our body, we just bother about reacting because we feel that we have no option to take without taking this. We have to take, and resulting in huge problem. Before that, I just want to tell you, many of you can feel what is the problem if I take some additional drug molecules, what will be the problem? Just think of the particular number, Avogadro's number. 1 gram mole contains how many molecules? 6.023 into 10 to the power 23. Unthinkable. You cannot, if you write it on the board, you cannot read it. It's my challenge. It's very difficult to read it. Just write on the board, blackboard or whiteboard or green board, whatever. You will find it's a huge number of molecules. So if you take few milligram additional or extra, what you call extra amount of drug, these molecules are not your friend. They are going to your system, means they will definitely do some harm. So you see, any additional amount of drug if you take, this is definitely be harmful. In our time, little earlier, I think even about 10 years back, we used to take paracetamol 500 milligram only. 500 milligram, which is good enough. It used to work. But nowadays, you see, we're taking 650 milligram, we're taking additionally 150 milligram. This additional 150 milligram drug contains how many molecules you calculate. And imagine those molecules are going additionally without any reason. They are harmful. Paracetamol has bad effect in liver. So paracetamol has no dose, it has a very good, amount of tolerance. It is considered to be safe. 4 grams per day will not create much toxicity in your body, but it has a problem in liver. And that's why paracetamol is never given as a dose. It is given as SOS. When it is required, you should take that dose. Like 3 times a day, 4 times a day, paracetamol is normally not prescribed. It is considered as SOS when it is required. Anyway, I'm not talking about only paracetamol, any drug. any drug, any additional amount of drug molecules if you take, they are not your friend, and definitely they will just do something harm to your body. Next thing I want to focus that, okay, fine, we at this stage, we understand that additional molecule, extra amount, whatever we are consuming, which is not required, if we can locate the drug to the proper location, you know, that would be good for all of us, but we are unable to do it. That's why we are taking, consuming more drug and causing lots of harm in our body. So this is the purpose. This is the desired thing for drug targeting, so that we can consume less toxic materials, drugs, all, additional things we can get rid of. We can take the amount of drug which is just needed, so we will get less toxic effect of the drug, less bad effect of the drug, and additionally, that we can be safe in terms of, drug consumption. Second thing is, that we are delivering it with the drug vehicle, that's targeting vehicle. That vehicle is here the nano size. Nano size, you understand. Nano means small, very small, and it's a scale. Millimeter, micrometer, nanometer. So thousand, so it's very small. Then picometer. There are, this is a scale of size. So nanometer, why it is special? Why millimeter is not special? Why micrometer is not special? But why nanometer is special? Because nanometer, when you are going to the nano dimension, you are getting some advantage. So as you are getting some advantages, that's why it is special. What advantage you can get? Many, many advantages, unthinkable advantages. One thing, one example. The example is the material which, or you can say the drug, which is not, penetrating, passing through the cell membrane into the cell, that will go inside the cell. For incorporation of drug into the cell, it needs receptor. So if the receptor is not there, the drug will not go inside the cells. And many drugs are rejected, simply rejected, because in outside, outside it works very, very nicely. But when you are giving into the body, it will not work properly, because it is not going inside the cells. It's not reaching to the receptors, because that receptor may not be there. Now, what happened, not only receptor, other things are there, but, like binding proteins, distribution, everything is there. So important. But in this particular case, what we are saying, that the material which was unable to go inside the cell, it will go inside the cell. Because of the size of the material, it has, it changes lots, it brings lots of new physical properties, like supermagnetism, like the material, which was not supposed to dissolve in water, that will be dissolved. Very interesting. The things which are not supposed to dissolve in water because it has solubility problem, that will be dissolved because of the nano size. Nano size means very, very small size. You can imagine one atom is 0.1 nanometer. 0.1 nanometer. One protein size is 4 to 10 nanometer. The smaller protein. Larger proteins are even larger than that. Not only that. So from this dimension, bacteria, you understand, we cannot see it by naked eyes. Its size is up to 1 micron to 10 micron, up to 10 micron, larger, much, much larger. So nano size is very small. And I was telling you that because of the nano size, we can get lots of changes in the physical properties. Like the physical properties I have given here, you can see size distribution, agglomeration. It can form the colloidal solution also. Even the solubility, simple solution in water, and also crystallinity. Then, you know, the many other things, this sort of properties like chemical properties, also some cases, chemical properties, physical properties changes. If you can take the benefits of it for your formulation, obviously you can prepare nano size formulation. Now I was little earlier, I was telling you that because of the nano size, it can give lots of miracles, lots of things, important things. You know, gold. You may have gold ring. Those who are married, they have gold rings. Although I don't have. I am a married guy. But I don't believe in putting ring and all these things because I believe in my heart. So anyway, but many of you have rings. These rings, you know, made of gold, and you know, it doesn't get dissolved in aqueous media, and it is dissolved normally in aquaresia. It's a ratio, 1:3 ratio of hydrochloric acid and nitric acid mixture. So there only it can get dissolved. But if you bring this gold into nano size, no aquaresia is required. Simply because of this size, it can form gold solution. You see in the picture, there are different gold solution with different colors. So even depending on the nano size, you can get different colored solution of gold. It could be watery, it could be blackish, reddish, little oranges. So different type of colors can be obtained simply because you are converting into it nano size. So that is the advantage. If you can bring the material into nano size, you can get several important changes in its property. What we are going to do, I will here focus few drug targeting approaches because it's a short time, you understand. So here, basically, we will discuss about 3 drug targeting approaches, how we can go for drug targeting. First approach, here we are, what I am going to present is with Aptamar. What is Aptamar? Aptamar, basically, they are the small nucleotide sequences, DNA or RNA. You know DNA, RNA? DNA, RNA, basically, they are having, you know, nucleotides, and basically they are connected with sugar phosphate linkage. Linkage is very important. So sugar phosphate linkage, and this is, you know, the Nick sequence. Nick means small single-stranded sequence. So does it mean that every small single-stranded sequences are aptamer? The answer is simply no. All single-stranded DNA cannot form a 3-dimensional structure. Aptamers are those single-stranded nucleic acid sequences which can form a 3-dimensional structure. And that's 3-dimensional structure often unique to bind with some proteins. So this is important. If your aptamer, whatever you have selected, and that aptamer has a 3-dimensional structure, fine, but it is unable to bind with the desired location, it will not be fruitful for drug targeting. So it is important first to just start with lots of aptamers. Then we have to find out which aptamer is possibly binding, and then you have to go ahead with that aptamer. This work is just a good, you know, materials for learning, those who are willing to do or willing to work with aptamer in future days. So basically, aptamer is the nucleic acid sequences, either DNA or RNA, doesn't matter. But the main thing is very important. What is the main thing? Main thing is they are, you know, basically form a 3-dimensional structure. And that 3-dimensional structure can bind with the desired location. Only then it will be fruitful. So it is like, you know, you have to, there is no ready-made available thing. You have to try. You have to try. You have to find out that, okay, this aptamer could be able to bind. And then you have to study, you have to work with that, and you have to select that which are binding. Okay, so this is a long approach. As you know, most of the cases in research, you have to do it. Then important thing is, in case of aptamer, you see, you are binding it with the nanoparticle surface so that it can go and bind to the right location, and it can deliver the materials drug from the nanoparticle into the cells. But basically, aptamers, as I said, they are the single-stranded nucleotide sequences. Single-stranded nucleotide sequences means what? They are basically, what? They are having a problem of destruction by nuclease enzyme, like, basically DNAs, RNAs enzyme. So, because they're single stranded, so it is essential to protect them. How can you protect them? You cannot change the basis because adenine, thymine, cytosine, guanine, or uracil in case of RNA, you cannot change these bases. If these bases are changed, there will be a mismatch. There will be perfect, structural problem. You cannot change the sugars because sugars are important, like deoxyribose sugars are actually for DNA, ribose sugar for RNA. So then where you can change? You can change in this sugar phosphate linkage. DNAs, RNAs, they normally work here. They split the nucleic acid here in this region. So, there are oxygens. So you are replacing this one of the oxygen by certain other compound. Normally, we replace this with sulfur. It is called phosphorothioate. So when you are using phosphorothioate, when this single-stranded nucleotide is passing through blood, it will not be affected by DNAs or RNAs because DNAs, RNAs, they don't have eyes. They can only recognize the sequence and then they split it. So since they're unable to recognize the sequence, you have modified it, so they're unable to go and split the nucleic acid. So nucleic acid, even single-stranded, remains safe in blood. So that is the objective. And that's why I said little bit idea about aptamer and its use. Now this work, because once this presentation is over, you may have the curiosity to get the material and about the work. This work has been published in 2020 in June, very recently, we understand, in a very reputed journal, Molecular Therapy Nucleic Acid. So this work, those who are interested, this work is freely available. That is the journal. From journal, you can download it by yourself. So there's no problem at all. You can download and you can just investigate or use its, different scientific advantages if you want to continue this study. Now, okay, all the details, whatever is there in the paper, it is not possible to focus because you understand we are running with the time, but some important data, some important information, we are just sharing. These are the nanoparticles. Might be you are well aware of this polymeric nanoparticles, and maybe before my presentation, somebody else already have given such presentation regarding polymeric nanoparticles. So details of its preparation and other things I'm not going to discuss. This is there also in the paper. I have just given the reference. So this polymeric nanoparticles, once developed, you have to, as usual, you have to characterize it. Characterization, by this size, save, zeta potential, and all these things. So even before that, drug polymer interactions by FTIR. So all these studies are there in that paper. If you go, you will find these things. So I'm not going to discuss about this thing. I'm going to discuss about some fundamentally important thing related to this work. So you see, this polymeric nanoparticle, this is the same picture, scanning electron microscopy. It produces the surface area, but this is FESM. Also, it gives some idea of the three-dimensional structure, and they're circular in shape, more or less. This is stem. You see the drug distributions are there. It's more or less scattered drug distribution. We use paclitaxel. These are, AFM. So it's uniform size. The most important thing now I'm going to discuss, this is very important, that aptamer conjugation, whether the aptamer has been conjugated or not. So for that, we have used different aptamers. But here, two aptamers I have shown, the most important two aptamers, which we selected finally. One is aptamer 5, we have named it L5, another is aptamer L2. So you see, here the most important thing is, these are the well. In well, you see aptamer conjugated nanoparticles are there, because nanoparticles could not move in the gel electrophoresis. But aptamer, you see, here you see, this is one, and this is another. This is smaller size, this is little larger size. So you see aptamer, these are the ladder. These are the ladder to understand its size, or how exactly whether this aptamer size is matching or not, etc. So basically, the aptamer conjugated nanoparticles are there in the well, and the aptamers are actually free aptamers are moving. So in the different lanes, so it is understood that aptamer has been conjugated in the nanoparticles. This is very important, that whatever we are working, you have to understand that it is perfectly all right or not, because it's important to understand that I have conjugated aptamer, whether it has been conjugated at all or not, because this is nano size, you cannot see it, and that's why this type of studies are important. Now you see, then the uptake. Well, I'm telling you, all the studies, whatever we have done, it is not possible to describe or discuss today at this time. So I'm just going to discuss about very important information. So one thing I said, yes, we have developed aptamer conjugated nanoparticle. Then we have gone for drug content, drug release, type of drug release, without aptamer, with aptamer, all these things have been done. It is there in that paper, you will find. Then important issue is that okay, our aptamer, I just want to tell you one more important information. We have not done simply with aptamer also, then we have used two other proteins also. One is transferrin and another is galactosamine. So these two proteins also we have used for conjugation purposes. Anyway, but they were not very successful, so we rejected. And we selected the 5 aptamers, and with this is, the control group has no treatment at all. And then this is aptamers conjugated, this paclitaxel nanoparticle without aptamer, and these are all paclitaxel nanoparticles with aptamer. So paclitaxel is our drug. And we wanted to see whether there are these, polymeric nanoparticles are taken up by the cells or not. This cellular uptake is a very important study, in vitro study. You have to do it outside the body. So we have to take at least 2 cancerous cells, at least 1, normal cells, normal hepatocyte. This study is on hepatocyte, you understand. Our target is liver. So we have taken 2 cancerous hepatocytes. One is HepG2 and the other is HUA7, HUA7 cells. So these 2 cells are cancerous cells. And we have also used normal cells also, normal hepatocytes. And now if we compare these findings, you see, in all cancerous cells, these nanoparticles have been taken up well. Just check. All the cancerous cells, you see, they have been taken up. Also, the uptake is variable, but they have been taken up. And you see L5, just see, these has been done by FACS, flow cytometer. So, you know, this is one of the sophisticated instrument which cost about 2.5 crore nowadays. So this data is very, very authentic throughout the globe. So you see, this has been taken up. L5, that is Aptamar 5, its amount is much, much greater as compared to control, that is normal, cells, control cells, then Aptamar, normal nanoparticles without aptamers and the nanoparticles with aptamers. So all these treatments, but here distinctly you can say you are getting L5 and L2 having much better result as compared to any other group. So you see, if you compare this thing, you can understand that the paclitaxel nanoparticle conjugated with Aptamar L5 and Aptamar L2, they have been taken up in a much greater way as compared to the other cells. Then we have gone for the normal, you know, determination process of IC50 and different formulation. There also you will find that L5, this is L5, and this is L2, they are very potent. They are very potent. This is very interesting. This HepG2, sorry. This is HepG2 and HUA. See here, there are 4 cells. One is Chang liver cells, another is WRL68 cells. These 2 cells are normal cells, normal hepatocytes. And HUA and HepG2, the cancerous hepatocytes. So you see, that is carcinoma cells. So you see, these cells, you see the results are absolutely much better for L5 and L2 as compared to any other. PF is the marketed formulation. So we used one marketed formulation, paclitaxel marketed formulation. So if we compare, even in all the cases, you see L5 and L2 are quite potential. Then we have gone for the Chang liver cells uptake results and also WRL68. Here, we got some interesting findings when we have gone for flow cytometer analysis, flow cytometric analysis. You see here control, just check control results and the L5 and L2 results and others are more or less same, So aptamer conjugation doesn't create any problem for the normal cells. So its uptake was not modified by aptamer conjugation. So here, you see, here this finding is striking us. It gives a big, you know, what you call the striking. Yes, this aptamer conjugation, they are allowing the material to go inside the cancerous cells in a much greater way, whereas this is not just doing the same thing for the normal cells. So this is the first finding where you can just look at, yes, it could be good for successful drug targeting. So here you are getting first initiative that results are actually going to help us for drug targeting. Then we have just gone for other studies slowly. Here, you see, this is another very important study. This study, basically, you understand the cell cycle analysis. Cell cycle analysis, we have analyzed the cell cycles in the cancerous hepatocytes because they're very, you know, proliferative. They the proliferation rate is very high, they're cancerous. So basically, you know, we studied the cell cycles. What you are getting in L5, and in L5 you see the ratio of G2M, G2M, G2M you understand, G2 is, okay, let me a little bit just for the recapitulation. There are 4 phases of cell cycle if you consider. Basically, S phase, here DNA synthesis starts, cell is getting ready for the division, right? So DNA synthesis is started. Then G1 phase. G1 phase, here the DNA synthesis and protein, RNA synthesis, protein is eventually. Then G2 is just, it's a growth. It's just cell is almost ready for cell division. And M is, you know, is for mitosis. That means cell division started. And paclitaxel, you know, it works in the mitotic process because it's blocked the spindle for just stopping the cell division. So you see, if you go for the cell division, cell cycle analysis with these facts, you are getting this analysis, the data. You see, here also L5 strongly working on G2M phase. And so similarly, you will get it for the L2 also. So the findings are quite, you know, interesting. Here also we worked with different formulations, including this protein conjugated formulation, but none of them are very potential comparing to this L2, L5 and L2. Then we have selected the DNA fragmentation study because M2, you understand that this is the mitotic cell division. So here, the cell division process, if it is there will be, and you are blocking the cell divisions, and that causes lots of fragmentation in DNA. And that is true, actually, you know, when we go for, all the studies have been done by, you know, what you called is the FACS, that is flow cytometer. So here, ApoBRD DNA fragmentation assay has been done. And here also you see just see the figure. I do not know whether you can see it or not, but here you see there is a huge difference. This is 86877 is its data, whereas control is 3987, even in 4 figure and 5 figure. You understand that how much it is actually causing the DNA fragmentation and not allowing the cell division. So this is actually, drug is capable to work very strongly at the mitotic cell division process. Now here, what we understand at this stage, that yes, everything is fine, drug is working at the right phase of cell division, it is confirmed, it is able to go penetrate inside the cancerous cells, it is not going to the normal cells. So these data are quite satisfactory to go ahead for the further studies. Further studies means, you know, the apoptotic studies as well as, you know, the biochemical and morphological studies. Now here, you see, first I can show you the apoptotic studies. Apoptotic studies, you know, this is one of the very important studies, and acridine orange, ethidium bromide is used for this purpose. You know, acridine orange and ethidium bromide, basically this staining, what it does. This is very interesting. How will you understand the cells are apoptotic? And what type of findings are there? Apoptotic cells means basically you will find that there will be the cellular shrinkages. Then also you will find membrane blebbing. Then also you will find, this is very important, chromatid condensation, particularly at the perinuclear areas. And also you can say formation of apoptotic bodies. So these are the things which is normally, the marker, or which you can say the parameters by which you can say that yes, these are the apoptotic cells. Also, there will be some patches type of things in the apoptotic appearance in the cells. Now, what happened, that initially, if the cell is normal, you will find the green color appearance. In the early apoptotic cells, it will be much brighter green. So green, normal. Brighter green, this is, early apoptotic cells. Because here in the early apoptotic cell, what happens, acridine orange is able to penetrate into the cell membrane because of the, some uh, variation in the membrane, what you call is membrane integrity problem. So in membrane integrity problem, acridine orange goes more, and this apoptotic bodies and these things, they are actually, they are brighter. So early apoptotic stage, this is also green, but it's the things are brighter, and you will get the peripheral nuclear, the chromatid condensation and all these things. Then late apoptotic orange to red. But here the appearance are all those appearances are there. Chromatic condensation is there. Then what you called is this chromatid condensation, then the apoptotic bodies, and these things are regularly a common parameter to observe in case of the late apoptotic process. Then necrotic process. How can you understand the cell is necrotic? Cell is necrotic. Here also it will be orange to red. But only difference is there that here the cells will be absolutely like normal. Absolutely like normal. No chromatid condensation, no apoptotic body form. It is like normal cells, but red in color. But so I have given you 4 stages of this process. First stage is live normal cells. Live normal cells, green in color, nucleus is there, no such chromatid condensation is there, no membrane blebbing, no, that is the loss of membrane integrity. All these things are not there. So this is normal. Brighter green, chromatid condensation, perinuclear chromatid condensation, green patches, all these things are there. This is early apoptotic. Late apoptotic, then the color changes. This is from orange to red type of color. Here, all these things are more, you will find this chromatic condensation, apoptotic body formation, and all these things. And necrotic cells, they're red, orange to red, but their appearance is absolutely like normal cells. So these are the things. Question is, what type of findings do we like? We don't like necrotic appearances, because necrosis means it indicates the toxicity. It indicates the cellular death is because of the toxicity. So necrosis in your finding is never a fruitful finding. I can say this is a finding when you will achieve, you will get, you have to be careful. You have to change your formulation or drug content or whatever, because it shows that your formulation or your drug or something is very toxic, at least at the stage where you have applied. So necrosis is not wanted. You see, in our case, if you see MF, said marketed formulation, you can see lots of early apoptotic stages there. But you see PTX nanoparticle and PTX with L5, L2, and L1, there are late apoptotic cells also. Huge late apoptotic cells are there in L5 and L2. Even in L1, you see, there are few, but there are mostly necrotic cells. So we rejected L1 and we have selected for L5 and L2. That was the results of, HepG2 cell, cancerous And this is the results of Hua cells, another cancerous thing, cells. And here also the finding is more or less similar. You see L5, L2, they're showing lots of late apoptotic cells, whereas normal nanoparticle and also the MF, that is marketed product, marketed formulation, they're actually showing some late apoptotic cells are there in Hua, but also many of them are in the early stage. Now, if you go for the normal one, the cancerous one, if you compare with the different formulation, at a stage, the same figures, it looks like that. Here you see, we have given the data of the protein conjugated nanoparticles also. This, yeah, you will find. So the findings are not very satisfactory for the others except this L2 and L5. Then we have gone for, mitochondrial membrane potential. Mitochondrial membrane potential has been done using Jc1 stain. Jc1 stain, it gives you two ideas. One is the cellular mitochondrial membrane potential and whether it is lost or not. And the other is, the different apoptotic processes or necrotic processes. So you see, A, B, and CD, both of them are for A for HepG2 cell and Hua cell, and CD also for HepG2 and Hua respectively. Now, first part, you just look at A and B. A and B here, you see, what we have done, different treatments are there. You see, it is written there in every box. Just see the cursor, here it is L5. We are interested in L5 basically, and this is L2. Here also you will find L5, and here you will find the L5 and L2. Here it is L2. So what is important here, Jc1 staining, it is what? It is basically Jc1, it's a monomer. Jc1 staining is a monomer stain. Now you see, when it goes inside the cells, and if they aggregates, they form red in color. Monomer gives green color. When they aggregates, they form red in color. So you see, so green to red, this ratio, or rather I should say red to green ratio, if it is more, that means more going towards red, that means mitochondria is losing its membrane potential. That means cells are dying. Cells are dying. So again, if you just look at this picture, I have shown already the places like L5, and this is L5 position, this is L2, and this is L2 position. This is for HepG2 and this is for Hua. You see, the percentage is eventually increasing. Mitochondrial membrane potential is getting lost. Basically, in case of the control cells, and there, eventually these ratios are, enhancing. Basically, when the rate is enhancing, mitochondrial membrane potential is getting lost, indicating that the cells are actually dying, but its effect is more or less towards, necrotic cellular division. So, that if we don't want, I said that this process should be through the apoptotic one, not the necrosis. You can see many cases, here you see, like VF, marketed formula, is going towards the necrotic cell. Here the four chambers, the first one live, second one we indicate, this is for the control, but first one is for live cells, second one early apoptotic, third one late apoptotic, and this is necrosis. So you see, normal case, no early apoptosis, no late apoptosis, normal cells to necrosis. Although here the normal cells to necrosis is, I think, 12%. Here you see But L5, you see, it goes through normal, then early apoptosis, late apoptosis. Whatever we have found in morphological study in the cells, it is just mimicking, you see. The huge late apoptotic cells are there. So this is also here, you will find a similar finding. And that is true for L2 also. See, it is going to late apoptosis and huge late apoptotic cells are there. So that findings indicates that yes, this is basically, beneficial, and we should go for the further studies to understand it. Then we have gone for the reactive oxygen species. Reactive oxygen species, it's very important for cancer study. And this Ross analysis, once again, you see, shows that L5 data and L2 data, you see, compared to control and also, the other marketed formulation here, Ross development is quite high for the cancerous cells, because by this, they're trying to destroy the cell. So Ross enhancement in cancerous cells is actually beneficial to kill the cells. Now, if we go for the controls, that is Chang liver cells and WRL68 cells, these are all normal liver cells. You see, except marketed formulation and this protein conjugated formulation, nowhere any much change is there. Yes, as I said, necrosis was taking place by L1, so we have already rejected these three. And you see the other cases, L5, normal nanoparticle and control. Here we have more precise, because we have selected L5 based on all the data, because L2, L5 was good, but L5 is more potential. So we have rejected at this stage L2, and L5 we have taken, compared with others. And we found that in normal cells, L5, normal cells, and normal nanoparticle treated cells, they are more or less same. So this was quite satisfactory in every, types of data which favored us to go for in vivo study. So we have the basis of all these things, whatever I have discussed, it is given here in nutshell. Now, we have, at this stage, we try to focus on the targeting in the in vivo model. Whatever we have done in vitro. In vivo study, where this is the study we have, because nowadays lots of limitations are there. You have to develop the animal model properly. Numbers of animals also are very limited. They don't allow you to go give lots of animals to work with. So we have selected 8 groups. 8 groups is the first one is the normal group. Normal group means they just receive water as well as this food, nothing else. Group 2. Group 2, obviously, carcinogen treated group. Carcinogen treated group means cancer control. That is, cancerous process has been developed in animal model using carcinogen. And then carcinogen, how long we have treated? From 3 weeks to 20 weeks. What carcinogen we have given initially? We have given, DENA, diethyl nitrosamine, 200 milligram per kg body weight, 1 IP injection, followed by 2AF, 2-acetylaminofluorine. It's a very potent liver carcinogen, and we used it 0.5% weight by weight with food. Initially, it is given mixed with small quantity of food in a cage, and then after 7 to 8 hours, we used to give them the normal food. Overnight, normally they starve. And then in the morning, they used to be given this carcinogen mixed food, and again 7 to 8 hours after the normal food. This was the cycle for the treatment of AF. And the other thing you understand, the group 3 also carcinogen treated rats, treated with marketed formulation. Group 4, This is just carcinogen treated rat received the paclitaxel loaded nanoparticle conjugated with L5. Group 5 is simply without L5 conjugated nanoparticles containing paclitaxel. And group 6, obviously carcinogen treated animals received paclitaxel loaded nanoparticles conjugated with L2. Here L2 we have also included just to see its potential, and then finally it is carcinogen treated rat received, you know, the PF. So you can understand that that is in all the cases the model is up to 23 to 20 weeks. This treatment was there. Then there was a cap just to develop, allowed to develop cancer up to 36 weeks. And then around 38 weeks, we have, killed the animal, and the treatment started from this period to this period. So it is given here, all these markers are there by which you can understand that the treatment, how long we allowed for treatment, etc. All these cases, let me tell you, IV injection was there through Telman. So this was the entire process of animal model. And then, once at around 38 weeks, once we kill the animals, and then we have taken the liver, and the livers have been studied for different enzymology, different marker proteins, as well as histopathology. These are the histopathologic slides. You can see the liver lesion. You might have studied, you might have seen, always studied in liver lesion in liver cancers. What are those lesions? This is the lesion. You see, this is ground glass lesion. It looks like ground glass. And this is a basophilic lesion. The tolerant blue slide, and this is, these are very interesting. These are not, void. They're cells. These cells, you can see, every cell has a pointed nucleus. Pointed nucleus. Sometimes it is more than one, and they have, a peripheral arrangement. That means these nucleus are there at the periphery nuclei, and these cells are called spongiosis hepatis. Spongiosis hepatis, basically, they are the precursor cells of liver cancer. So if these cells are ever seen in any histopathological slide, you can say the man is just going towards cancerous process. So this is a kind of clear indication to understand that the process is on to develop the cancer. Now, if you go for the other cases, other slides, you can see this is with hematoxylin, and here the hematoxylin eosin slides. Here the change, the improvement after the treatment, eventual improvement you can see, you can notice how gradually and eventually the process is improved. This was in the first one, you see lots of necrotic cells are there. It is getting removed. Eventually, you see the changes here in the cancerous process. You see, there are lots of, what you call the sinusoidal space between the hepatocyte lineages are there, but this is changed after the treatment. How much it is changed, you can see, and that clearly demarcate, that can give you the idea that yes, this treatment, if you go for targeting, it will be very successful. Here we have given some data, quantitative data, that in every case we have taken 6 animals, and you see, if you go for this treatment area, there is no tumor was obtained, but in other cases, you see tumors are there, cancerous tumor process are there, but here tumor is not there in these 2 cases. And the lesion size is also a big matter. Smaller size lesions are more in these 2 cases, whereas larger size lesions are much less. So that means it delays the cancerous process. Cancerous process is slowing down here. So that is a clear indication of this quantitative data. Now we have gone for the apoptotic studies, apoptotic marker proteins we have selected, and we wanted to know that whether they are really changing the apoptotic or modulating the apoptotic protein. And you know that yes, it indicates, it clearly indicates that the changes or modulation of apoptotic protein uh, is there by the treatment which favors to kill the cancerous cells. Then also we have gone for the bioavailability of the formulation in liver as well as in blood. The data is there which shows that you see the bioavailability, more bioavailability is there in case of the nanoparticle formulation than the free drug as well as the uh, the marketed formulation. So it is understood that obviously nanoparticles will be present for a longer time in liver. This is the data of the normal liver undergone treatment with this, and this also clearly indicates that their uptake in liver is not there. That's why the cells, liver hepatocytes are actually, it looks normal, and that favors the investigation further. Now, if we go for the biochemical parameters, these are the different enzymological studies, common enzyme, SGOT, SGPT, and alkaline phosphatase, etc. So this indicates that yes, the treatment has no much liver toxicity in normal animals. Even you can say in normal animal, normal control animals, and also in by any of these treatments. So except some this formulation and free drug, they are having some toxic problem, but not that very significant in case of the Aptamar formulation. So here what we find that L5 shows maximum potential in case of the treatment of liver cancer. Now, fantastic. After this is we understood that we have developed a formulation. It was conjugated with L5 and it works quite nicely. It actually shows very potential findings. Now, question is, these aptamers are binding to the liver cells, hepatocytes, on the membrane. Where? Where is the location? Where are they getting bound? So we have no data. So we have gone for, obviously the homology model by docking, and we wanted to find out which proteins actually are there going to bind. And this study, you see, we have found that two proteins are actually, they're going to bind with two proteins, L5 and L2, and the binding potential, binding data are there. You see, one is heat shock protein, and the other is, TAG72. So these two proteins are actually getting bound. GAG is tumor associated glycoprotein, 72, and heat shock protein. So these two proteins are getting bound and with this Aptamar. So this was the findings of the entire study. Now, let me tell you about the study. One more point I want to highlight, that now we are actually investigating in the real terms whether these proteins are getting bound with the aptamer. And yes, the findings are also favorable. Now, you see, but since we are yet to communicate it, so we need to wait for some time to deliver this data. Now, you see, this is another, because I have the time is till 1:30. I will just discuss it a little bit faster. This is also a very current findings, current data of ours. This case, we have used CD340 monoclonal antibody. It is just with the nanoparticle we have conjugated, and then the particles containing the doxorubicin drug, doxorubicin. And the purpose was to, obviously, to induce the apoptosis, that means to reduce the tumor. And the second one, to reduce the cardiotoxicity, because the drug doxorubicin causes cardiotoxicity. So this has been published in the International Journal of Nanomedicine. Those who are interested, you can select that paper. And how we have just conjugated this antibody with this nanoparticle, we have used this entire chart. It is there available in the, basically in the journal. You can see it. What we did, basically we have used BS3, it is subbaric acid, bis subbaric acid. So this has been used. And we have used also TPGS, tocopherol polyethylene glycol succinate. So these two materials have been used to conjugate the nanoparticle. Entire procedure is available in the study. This is the method how we have conjugated antibody with the surface of the nanoparticles. Now these are the physical data is for this nanoparticles, the size, zeta potential, etc. This is for the stability data. Now this is also related to the SEM, TEM, etc., AFM. So I'm not going to discuss much about this. This is the release study, and that kinetics pattern. Now here, important slides is that here you see you have to show that your nanoparticle has been conjugated with the aptamer, I beg your pardon, with the antibody. So we have given 3 different methods to prove it. First one, you see, this is the gel electrophoresis, vertical gel electrophoresis. We have conjugated FITC, we have used FITC conjugated antibodies. So obviously, in the gel, when we have taken the picture, this gives green color. And this is that's normal gel. Here we have run. You see this case, antibody conjugated nanoparticle is there, and this is free antibody, and this is nanoparticle without antibody, so it has no, obviously, no protein available, so it is not there. And this study was also done by FACS analysis, and we have shown that yes, antibody conjugation is there. Once this is proved, and then we also, these are the stability data. I have told you a little early that they are stable, the formulation is stable at 4 to 8 degree. In other cases also it is stable, but the problem is at higher temperature, like 40 degree, 75% humidity, the polymeric nanoparticles little bit, soften, and kind of coagulation type of appearance is there. Now MTT assay, MTT data, these are all standard. I have described it a little early. We have used 3 cells here, SKBR3, MDA MB231, and the other one is, MCF7. Here, this is very important, that why we have taken these 3, because our antibody was focused particularly with HER2, HER2 receptor. So it will bind with HER2 receptor. So we have selected these 3 cells because SKBR3, it has a very high level of expression of HER2. Whereas MCF7, it has moderate, and MDA MB231, it has no expression. So we have selected these 3 cells because of that. Then we have also seen the uptake. Here we have used DAPI. DAPI is for nuclear staining, blue color, and doxorubicin itself, the drug itself has fluorescence, red in color, and FITC we have given. So all these things here, green appearance shows the FITC for the antibody. So you see in SKBR3 antibody conjugated nanoparticle availability is there. But you see in other cases like MD MDA 231, it is not there. So that indicates that conjugation is successful and antibody is going to the cells where the receptor is expressing. Then we have gone for the other analysis, like whatever I have shown you little early, like cell cycle analysis and others. So here also, the cell cycle analysis shows that percentage of different percentage of cells, how much doxorubicin and nanoparticles and other, how much it is going inside the cells. This is, and what type of cell cycle phase is arrested, and it is just basically mimicking the follow-up of doxorubicin. And then apoptotic cell division, also apoptotic marker proteins we have investigated, and it favored that, data favored that apoptosis is there in the cells, MCFs. And this is for the in vivo pharmacokinetic study. This is the standard study. What I want to just show you here, one interesting data, just see the changes of half-life. This is the doxorubicin administration. This is doxorubicin nanoparticles. And this is doxorubicin nanoparticle conjugated with the antibody. They're more or less, you see, 27 or 32 hours, where it is 17 hours. So half-life, you are changing 10 hours means quite, 10 to 12 hours means quite a big change in the half-life. It will remain for a long time in plasma, obviously it's circulation. And that actually favors, similarly mimicking exactly the clearance data also you can find. Then, as I said, we wanted to see the, if we just incorporate it in the nanoparticle, whether it will cause the toxicity, cardiac toxicity, or it reduces cardiac toxicity, because our objective was to reduce the cardiac toxicity. And here the picture of first one is the RR interval, where you will find that cardiac toxicity, RR interval, you understand. RR interval, it gives QRS signal, basically, with the sinusoid nodes and also autonomic influences. It gives this RR signal. If it is faster or closer to each other, that means a heart, it produces lots of effect to the heart. So here you see the control, and this is the antibody conjugate nanoparticle. They are more or less similar, but you see only doxorubicin, it has enhanced. Nanoparticle also, it reduces, but not that, but control, you see how much, and blank nanoparticle, control and blank nanoparticle, you see they have no changes. Similarly, doxorubicin containing antibody conjugated nanoparticles, they also successfully reduces the RR interval close towards the normal. And if you go for the histopathologic study of the heart, here you will get a very interesting finding. See, doxorubicin, how much it is damaging heart. That means a person taking doxorubicin injection, it is how much dangerous for the patient, because its heart is damaging. You can see the within the same period of treatment, control and doxorubicin, and the control and blank nanoparticle, doxorubicin nanoparticle conjugated with antibody, they're more or less same. No much changes there, but huge changes there on free doxorubicin. So it is advisable free doxorubicin should not be taken or given to the patient. Otherwise, it will be very harmful for his heart. The other different data of enzymology related to the cardiac toxicity, and we have given LDH, other than that SOD also, superoxide dismutase, and then also tissue distribution, how much it is distributed in different tissues and organs. Obviously, liver should have more distribution in case of all studies, and kidney also should have. Normally it is because these are two organs from which materials are eliminated from our body. But if we go for this technetium level nanoparticle, and we have gone for this, imaging, and we found that radio level nanoparticle source with the hours, this is for 0.5 hours, this is antibody conjugated, this is without antibody, this is free-dark. And the changes are there, you see. But in case of this antibody conjugated nanoparticle, you see it is going towards the tumor. Interestingly, the free nanoparticle is also going towards tumor. But here, you see at least double, it is reaching towards the tumor. And then we also have taken out this tumor and then its size measured. As usual, data are there. You can see how much reduction is there in case of tumor by application of antibody conjugated nanoparticles. This is possibly my last study I will focus today, because I have 5 minutes is there. I will not take much time. Let me tell you, here we use docetaxel. Our purpose was to bring it to the brain. That is to cross blood-brain barrier. And here we have used liposome. You see, this is liposome. This picture shows liposome. Liposome, what is important, liposomes are normally very easy to prepare, and they always, they're more or less homogeneous in size. So you see, I cannot see whether you can understand it or not. This is, this is 100 nanometers. So these are about 20, 30 nanometer size. So 100 nanometer, you cannot hide anything here. It's very, and with the field division scanning electron microscopy. These are the size. You can say size is because when you are, that is absolutely, dried product lyophilized, and when you are determining its size, you have to disperse it in liquid, that is in water. So obviously liposome enhances its size little bit. So this size is, this is 10, 20, 30, 40, around 50 nanometer size is there. And here also, this is little larger. So different types of liposome we have used. We have selected this one. And then you see, this is a very interesting and important study. Liposome, when you are preparing, you have to understand these liposomes are not like solid nanoparticles. Solid nanoparticles is like marble. You know marble? It's like marble. But liposome is not like marble. This is like balloon containing water inside. Balloon, soft. So this structure is very important to show the integrity of the membrane. Integrity of the membrane. Your liposomes are remaining intact. And these are, if you want to do it, you have to use the cryotem. So cryotem, in that case, you can see the membrane integrity. If you look at this is 100 nanometers. So I think this is around 40, some of them are even less, 40, 50 nanometer. Here also it is around 50 nanometer. You see, membrane integrity is intact. In every case, no membrane is damaged. And here also, this is a bilamellar. Many in books, normally you will find the people says that below 1 micron, no bilamellarity is found. This is not true. In every case, we have found bilamellarity is there, even much below 10 nanometer, but 100 nanometer. But here, important thing is that this percentage is very low percentage. So it is like 1%, even below that. So this is there. Then we have just leveled it with FITC, and we sent it, injected it, and we wanted to see whether it is reaching in brain or not, and we found yes, it is reaching brain. So this is the first study where we found that it is reaching in brain. This is another very important study where you see we use the normal liposome and we have modified liposome. So we use this liposome And what we found, this is without the liposome, free drug. This is liposome. You see, it is reaching in brain. It is reaching in brain, but much less, much less. So what we thought, what we can do so that it can reach in brain much more. So I will show you this thing. You have, so we have used this simple lipid, DSPE. DSPE, you know, DSPE is a lipid which is available in our brain. So we thought, why don't you add some other lipid which is available in brain along with the normal composition. So what we did, we used this DSPE and see the miracle. See the miracle. The appearance of drug liposome in brain, it has at least 4 to 500 times more. 4 to 500 times more in brain. So drug targeting does not necessarily mean that always you have to go for the active targeting. Even some passive method can bring a lots of change of your formulation. It can, so thinking and how we can do it, this is very important. And if we can do it, it will be beneficial for us, for the patients, particularly the sufferers. Here we have gone for the brain concentration or kinetics of the drug. You see, the difference is of the two liposomes. This is the drug, and this is for the formulation with DSPE. The changes is there, you see. Similarly, in blood also, the change is there. This one is possibly another one. Here we have done similar studies, but here we have developed one lipid by ourselves, and that lipid, you know, worked nicely. This is also with the liposome. You can see the integrity of the liposome by bichitin, here also bilamellar. This is, so similar findings. So I'm not going to describe it much more. Just simply I want to show you that here you see it reaches in brain. This is our lipid, and this is, you know, the without lipid. This is not reaching in the brain. This is the normal liposome, and this is our liposome with the prepared lipid, and they showed more or less mimicking results. So how we can understand? Here we cannot understand. This is, you know, AZT. AZT, you understand? Yes? Zidovudine. Zidovudine is highly water soluble. It will not reach to your brain. You cannot send it. See how much it is going, and it is going in. We have improved with our lipid. It is 9.71, and normal liposome is 7.62. This is the half life, but the AZT half life is 1.15 hours. So imagine, even the highly water soluble drug can be sent in brain, and you can retain it quite a long time in brain. So these data are quite, you know, satisfactory. So far I have told you a few of our findings, how we can go for drug targeting approaches. And thank you for your patient hearing and giving time to me. Thank you very much. This is our university administrative building. This is our department. And lastly, the right side, you can see the stamp of our university. It was produced by Government of India at its 50th year. Thank you very much. Thank you all.
Thank you so much, sir. Near me.
Thank you.
Thank you so much. As always, your lecture brings so much wisdom, technical knowledge, and a lot of advanced current information happening around us with respect to drug targeting. Let me confess, sir, I heard you after almost 12 years. I found the same passion and same enthusiasm in delivering the lecture. I'm so fortunate and blessed to have you, sir, as my teacher. Dear participants, as we all know, right amount of drug at right time should reach to the right target of our body to get optimum therapeutic efficacy. Otherwise, we need to suffer from adverse drug reactions and undesirable toxicity of as it happens in conventional drug delivery system. You have very lucidly explained, sir, the complex technicalities of all the drug targeting by novel nano carriers like Aptamar, liposomes. Thank you so much, sir.
Thank you.
And sir, with your permission, I would like to raise some of the queries raised by our
Yeah,
participants in the chat
obviously.
box.
Yeah, but otherwise they can directly talk to me. I prefer to talk to them directly. They can, if you can allow them, because it is always better to talk to them. I prefer to talk to students or anybody front to front. It will be nice so that he or she can understand it properly.
Sure, sure, sure. We are trying to do that.
Mhm.
So could you please unmute the participants? Uh, one
You,
participants.
you can tell them one by one name, they can unmute and they can ask me the question.
Hello. Good afternoon, sir.
Yes, please.
Hello.
Yes, please. I listen.
Am I audible?
Yeah.
Actually, I'm Dr. Sushma Talegaonkar from Dipsiru.
Oh, how are you?
I'm fine. I'm good. Wonderful talk, as always. I listen here also in
Actually,
Dipsiru.
actually,
Yes.
I have lots of communication with you, but I have never seen you.
Okay. So
Can
should
you?
I
Yeah,
meet you?
you can. At least I can recognize you. Oh, I see. Good, 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 good.
Okay.
Thank you. Please.
So, sir, it was a very wonderful talk. Just one query.
Yes, please.
Even I wrote there also in the chat box. Sir, you have actually discussed about the docking, molecular docking of the ligands with the receptor.
Yeah.
So, just my question was sir, is it means what is the steps before first we should go for a docking, or should we have some experimental finding and then we can go for a docking?
I think it is always better to go for the experimental findings and then go for docking, because if you go for docking and if you select it ultimately whether if it does not work in the system, because docking always cannot give you the fruitful and correct result. Like we have gone for docking, we have got some results, findings, but I am not very sure that whether this docking is correct or not. So we have to justify it again with this protein. So what we are doing, we are using those proteins in vitro and then using the aptamer and trying to find out the binding and its rate of binding. And then once it is successful, as I said, we have got some initial results. As I said that it is successful. So docking is kind of, data which you cannot 100% rely. So first step is you have to find out whether the aptamer is working at all or not. Once it is working, then you should go for identifying the possible proteins, and then you have to do in vitro study with these proteins and its rate of binding, all these things.
But sir, experimental finding means the cell line studies or some chemical reaction? Means I couldn't get it. If I
Experimental
know.
study means, yeah, first of all, experimental study means first, once you have selected the aptamers, you have to bind with this, surface of this formulation. And once it is done successfully, you are satisfied, then you have to go for the cellular uptake in vitro. Absolutely. And then once you got the cellular uptake, preferably in normal cell, if it is not there, it is favorable. But if uptake is similar in normal cells and the cancerous cells, I think it is better to reject the data, because that data will not be very fruitful. So you have to always keep the target to study on at least two cancerous cells and two different normal cells. And you have to compare, preferably if your data is not affecting the normal cells properly, that means some surface proteins is there in the cancerous cells, which is actually aptamer can bind with that protein. So that is particularly, I think.
But sir, actually, I was thinking if suppose we have some findings means from the literature, and if suppose we are going for the first docking and then we will perform the cell line study. So we can save the time also and the chemicals and all everything we can save.
No,
Is it possible?
I see. The thing is that I'm telling you, if you want to work with Aptamar, major problem is Aptamar is a lots of reports are there, but actually when you will go to do it, you will find many cases because they have done it by docking, so
Okay.
these are not working. So
Okay.
because lots of actual wet lab study in Aptamar is not there. Whatever I found, mostly the Aptamar related studies are dry lab. They have docked the system and they have claimed that it could work, etc. So we have
Okay.
selected 8 to 9 aptamers, as I recall. Only
Okay.
2 aptamers worked. So you understand, remaining 8, whatever they have claimed is it did not produce results.
Okay.
So this is very important.
Okay.
But the way you have to select, selection
Okay.
of the way is depending on the
Yes.
person.
Yes. Yes. Okay. Thank you so much, sir. Thank you so
Thank
much.
you.
Thank you so much, sir. Any other participants? Yes, Shivani, you can unmute and ask your question.
Good afternoon, sir.
Good afternoon.
Sir, firstly, sir, I'm feeling very blessed to have a chance to interact with you, sir. Sir, because one reason is also you are also a guide of my guide, actually. So I'm pursuing
What?
my PG under Dr. Uttam Kumar Mandal, sir.
Oh, I see, I see, I see. Great, great.
Sir, I just want to ask, sir, how to determine drug amount within nanoparticles properly?
How to determine?
Drug amount within nanoparticles.
Yeah. See, if you want to determine drug from anything, what is important, you have to destroy that material totally to get the drug in the media. So you have to use these chemicals. Different people use different types of chemicals to dissolve these polymers so that the drug can comes out, and then you have to extract the drug from this mixture. So you have to, this is the procedure, and then you have to analyze. First of all, you have to take the polymeric amount of material, that is, you can say nanoformulation, you have to weigh it, and then exact amount, whatever you have weighed, that should be put in a container, particularly like, you know, what you call microtubes. And then you have to add, say, NOH or certain other solutions which can dissolve these polymers, and then you have to, the drug will release totally, then you have to centrifuge it number of times and wash it, and then you have to extract the drug totally, and then after that you can analyze, then you can say that, okay, from this much of drug is there from this much of nanoformulation. So you can calculate the percentage.
Thank you, sir.
Okay, one participant, Anis Pradhan. Anis, please unmute yourself and you can ask question. Anis Pradhan, is he there? So I think he is not here.
Yeah,
Hello.
Yes, yes.
Hello. So,
Yeah.
hello.
yes, please.
So myself, Anis Pradhan, and I'm from Sikkim. So my question is very simple, actually. My question is, what are the criteria for good nano carriers, sir?
What are the criteria of good nano carrier?
Yes, sir.
First of all, I think the size is very important. Secondly, is zeta potential. Thirdly, its drug content. Fourthly, is stability. And then you have to investigate its success in vitro, that is cell lines, and in vivo, in animal experiments. Once at those stages they are also successful, then I think you can bring this for further studies.
Thank you, sir.
One more thing I can tell you, that in animal study, actually 2 studies should be done. One is pharmacokinetic, and the other is, you know, pharmacodynamic. That is, you have to, in the disease model or whatever whatever disease or whatever the reason for which you want to develop this formulation. So that should be treated. But pharmacokinetic is equally, it should be done.
Thank you so much, sir.
Thank you, sir. Sir, just one request, as we have we are running out of time, so we will just take one question from Ashish Jain. So Ashish Jain, are you there? Okay, no issue. Is anyone?
May I?
Has...
Yeah.
Yes, yes.
Hello. Yeah.
Yes.
Good afternoon, sir. This is Bappaditya Chatterjee.
Please.
Very.
How are you?
Yeah, fine, sir. How are you?
I'm fine. Are you there in Malaysia or now right now in Kolkata?
No, I'm in Mumbai, sir. Last year I came back from Malaysia.
Good.
And in this COVID situations, I have heard you once back, few months back in one SPDS program also. So it's very nice. I'm hearing you, sir, the same voice, same tempo. Okay. Yes, sir. I just want to ask you actually some suggestion from you is when we work with nanoparticle related formulation developments, as you mentioned that stability and toxicity or adverse effect are two most important criteria. So what we usually do with the final formulation, we do check the stability and we do check toxicity in animal model or in cell line. So what do you suggest we should take this stability and toxicity testing inside our formulation development stage?
No, I should suggest first, when you are developing the formulation, more particularly when you have just developed the formulation, drug content and drug release up to this study, then you should go for the stability study. Because once we found it is stable, physically stable, then you should go for first in vitro toxicity study in cell lines. You have to determine this IC50 and all this things. And then if it is successful, then you can go for further all other studies, even including the in vivo toxicity. But first step is up to the stage of drug release, drug content, drug release study, then you should go for stability.
Okay, sir. Thank you.
Because I think that is the right stage where you should bring your formulation to understand whether they are stable or not.
Okay. Okay, sir. Thank you.
So any other thing? Any other question?
Sir,
yes,
can I take one more question?
Yeah, no problem. I should be happy. Huh?
Ashish Bhomik, are you there? Yes.
I don't mind.
Unmute yourself and sir, actually we have to start one more session at 2:30.
Yeah, there is no problem. But those who are having questions, they can send the question to me by email also.
That would be fine. Dear participants, sir is really approachable. You can have his email ID from the website, or I will update in the chat box or in our WhatsApp group. So if you need any other information, you can write to him. Sir is always there.
But somebody was going to ask me the question. Last question.
I think he is not here, sir.
Okay. But so, what was his question? You can read it.
Okay, sir. His question was, what does stand for notation of 7 in Q cell line and 2 in FG2 liver cell line?
I could not follow. Whatever?
What does stand for notation of 7? Sir, I think you have mentioned in your slide.
Like MCF7 and etc.
Yeah.
They are the different cell lines. Like MCF has different varieties of cell line, MCF7, MCF10. So depending on its nature, like protein expression, type of protein expression, like as I say, HER2, it is moderately expressed. There are MCF7 which are having no HER2 at all. There are MCF7 which expresses MCF, which expresses a high HER2 level. So it is like that. Similarly, different receptors, estrogen receptors, etc. Depending on that, cells are characterized. So all cancerous cells are not of similar type, even from the same origin, like breast, like liver. So depending on that, based on some character, these numberings are done.
Okay, okay, sir. Sir?
Yes, please.
One moment, sir. We have some thing to show you.
Mm-hmm.
Yes, sir, just as a kind gesture and the great sense of gratitude, we are presenting as a token of appreciation certificate for you. We will post this to your address. It's just a small gesture, sir, from our side for your contribution to this FDP. And sir, I thank you again for sparing some time from your busy schedule. We are highly indebted, sir. Hope to get your support in future as well.
Okay, first of all, I convey my sincere thanks and gratitude, and I appreciate to provide me this certificate. I wholeheartedly accept it, and I congratulate all the participants and also the organizers for your great effort. And but also equally, I feel bad because Bhatia is not there. Amit should be here today. But
Sir, actually, he is there just now in campus. Maybe, sir, he had some other engagement. That's why he could not fully attend. But he was there.
I understand it, but Amit should be there. Anyway, thank you. You know, Amit was
yes,
my
sir.
student also.
Yes, sir. Yes, sir.
So thank you very much. Thank you all, and wish you very, very nice time.